If you imagine your DNA right now, you'd probably picture that double helix you learned about in school at some point, hopefully, but it may actually look a lot different. Hey guys, I'm Sapna here at D-News. That ladder twisted on itself with bonds between 4 bases, adenine, guanine, thymine, and cytosine, that is the DNA double helix, the thing you were probably picturing in your head. But it's now thought DNA at times may have different configurations. The helix can twist the other way, have a couple extra strands. It can even twist onto itself into some pretty cool shapes. There are 3 biologically active types of the double helix: A, B, and Z. B DNA is that recognizable one. It has a right-handed twist, and it's the most common in living cells. A DNA has a wider spiral, and Z DNA twists the other way. It's a left-handed double helix with a zigzag structure. The biological purpose of Z DNA isn't quite clear. Since it twists the other way, it's thought to relieve some of the torsional stress on the structure. And some research shows it may play a role in transcription, where the genetic info is being copied. Beyond the double helix, new research says DNA molecules may also coil into a range of cool shapes called supercoils. Research published in the journal Nature Communications last October shows images of supercoiled DNA created by cryo-electron tomography together with supercomputer simulations. The scientists wanted to know how that DNA crams itself into a tiny nucleus, so they tried to look at it one twist and turn at a time. They found sharp bends, figure eights, even coils that look like handcuffs. Keep in mind this research, like most of the research on DNA, is in vitro or in the laboratory, but by using an enzyme called human topoisomerase 2 alpha, they did try to simulate conditions in the human body to show that supercoiled DNA is possible. So your DNA may be backwards or zigzagged or even twisted, but it might also have double the strands. Most DNA has 2 strands, but many people don't know it can also have 4. It's called Quadruplex DNA or G4 DNA. The G refers to guanine, one of those 4 bases that holds the DNA together and spells out our genetic code. G4 DNA is a topic that scientists are trying to learn more about, especially in the last decade, because it may hold the key to some major medical breakthroughs. Researchers in Sweden recently discovered G4 DNA in one species of yeast. They published their findings in the journal Nucleic Acids Research. Meanwhile, a team led by Florida State researchers looked for and found G4 DNA in the genome of the maize or corn plant. And in 2012, Cambridge University researchers claimed they saw G4 DNA in human cells. Using a fluorescence biomarker, they tagged an antibody they created to specifically look for the 4-strand DNA in human cells and bind to it. They found that G4 DNA seems to turn up most often during the S phase, just before cells are about to divide. Which brings us to the point of why we should care about 4-stranded quadruplex G4 DNA. If it plays a role in cell division, either speeding it up or slowing it down, scientists think G4 DNA may play a role in causing cancer and degenerative diseases in humans. So how do you turn it on or off? And is it possible to create a drug that targets 4-strand DNA and maybe stop cancer? We're not there yet, but that is actually a goal and focus for ongoing research. By the way, have you ever heard someone say, "I love to travel, it's in my DNA"? Is that true? Are some of us hardwired for wanderlust? Watch this video by Julian to find out.
The gene is also associated with people who like trying novel experiences and are hyperactive, and it's linked to ADHD. This variant of the gene makes people crave new experiences because it gives them a hit of dopamine, the neurotransmitter associated with reward.
So what do you guys think about 4-strand DNA? And do you have any other science questions you want to know more about? Tell us in the comment section down below, and don't forget to click subscribe for new episodes every day. I'll see you next time on D-News.